in general, these RNA binding proteins are probably going to bind mostly to the 3' UTR, the 3' untranslated region. And that's simply for two reasons. One is that if you think about the 5' end of the message or the open reading frame, these are constrained evolutionarily by their function in translation. The ORF has to maintain the proper coding region for the protein, and in both cases, the ribosome has to be able to pass through these regions. Okay? And that, therefore, any regulatory proteins which bind will be dislodged by the passage of ribosomes. And so this puts constraints on the ability of these regions to function as regulatory sites for RNA binding factors. In contrast, the 3' UTR really is free to evolve new structures, new binding sites, and factors that bind here are not going to be displaced by transversing ribosomes and can maintain their association with the message and therefore regulate its function in a more efficient manner. So how many factors should we expect to bind in? And we don't really know the answer to this question. The average, using humans as an example, the average 3' UTR is about 740 nucleotides, although it can vary tremendously from 60 to 4,000. Okay? So, but how many factors might you expect to bind here? Well, a low estimate might be 1 factor per every 100 bases. But we know that in general, proteins bind to sequence elements of around 10, and microRNAs bind to around 20 nucleotides. So maybe a high estimate would be 1 factor binding per every 20 bases, which would lead to 37 factors bound. Now this is probably unrealistic, but even at the low estimate of 7 proteins or factors being bound to the 3' UTR, you can see that the average message will have multiple regulatory factors bound to its 3' end, and therefore its control of localization, translation, and degradation will probably be a summation of the impacts of all these different factors, and not just the impact of a single sole protein. Now, a few other principles of these RNA binding proteins which are important to consider. First, they can affect more than one process. I want to go back to the PUF3 protein in yeast that we use as an example of a factor that binds and promotes RNA degradation. But we also know that this protein also binds and targets a subset of RNAs to localize to the surface of mitochondria. So this single protein then can both promote degradation and target RNAs for proper subcellular localization. And there are many examples of this where RNA binding proteins affect more than one process in the cytoplasm. Okay. Proteins associated with RNAs range from very general to highly specific. This is a diagram of the number of mRNAs associated with about 40 different RNA binding proteins based on co-immunoprecipitation from yeast cells. And what you can see is that some proteins, for example out here, the poly-A binding protein, which we would think of as relatively general, binds to approximately 1,000 mRNAs. And down here at the other extreme, some proteins which we think would bind to RNAs only bind to a few, less than 5. Okay? So RNA binding proteins can be either highly general, binding to many, many factors in the cell, or highly specific. And one thing we don't understand well at all is really what are the diversity of different RNP types and how does that affect the function of mRNAs in cells. But one thing we do know is that in many cases, RNA binding proteins then co-regulate mRNAs which have related function. So for example, using the PUF3 protein as an example again, the PUF3 protein binds to about 200 mRNAs in yeast cells. And essentially all 200 of those proteins encode proteins involved in mitochondrial function. Okay? And so it makes sense that they would actually, be coordinately controlled for their degradation and for their targeting to a specific subcellular location. And consistent with that, PUF3 targets these mRNAs to the mitochondrial surface and promotes their degradation under conditions where mitochondria are not as important in the physiology of cells, of yeast cells.